All right, welcome everyone, and welcome to the folks that are on Zoom too. That's why I'm using the microphone. So for those of you that don't know me, my name is Cheryl Logan. I'm a professor in the Department of Marine Science at Cal State Monterey Bay, and it is my great pleasure today to introduce Katrina, who's going to be defending her master's thesis. I got to serve as Katrina's primary research advisor through the Moss Landing program. And I'm going to spend a few minutes introducing Katrina, sharing her journey from where she grew up to how she got to Moss Landing, what it was like to have her as a lab member and as a member of the Moss Landing community, and I'll also share some of her amazing accolades as an academic researcher and teacher. Then I'll hand it over to Katrina. She'll be giving a 45-minute talk about her research. When she's done with that, there'll be time for Q&A. I'll ask for questions on Zoom and from the audience here. And then afterwards, myself and her committee will whisk Katrina away into a room where we'll ask her some more questions, and then she'll come back out here. So that's kind of what things are going to look like. So let's get started. Okay, I'm seeing Katrina's slides and not mine. Okay. I'll get it. Just there we go. Nope. Okay, so Katrina's parents shared some photos with me, and I think most of you know she grew up in Long Island, and as a young child, she loved animals. I love these cute pictures of Katrina here. Not surprising. She also loved the water and the ocean early on. Here's some photos of her boating and boogie boarding. And I did not know this, but Katrina also learned how to fish at an early age, and she was quite good at it. You can see there's some fish on the lines here. And she also just loved getting wet. And I love the expression on Katrina's face here because it just kind of shows her zest for life. Like, you can tell she's kind of annoyed, but she's like, oh, I'm going to get this person back. And that's kind of how she approached research as well. Whenever she faced adversity in her work and challenges, she always just like had a positive attitude and got right back up there and kept going. So that's something I really appreciate about Katrina. As many of you know, she also loves to dress up in costume. So it was fun to see that she was able to combine her interests in the ocean and marine animals and costuming early on as well. So this led Katrina to her degree at the University of Delaware. Her undergraduate degree was in wildlife, ecology, and conservation. She also minored in environmental humanities. She walked away with a 4.0 GPA from undergrad, which is pretty impressive. As an undergrad, she did spend the last year during COVID time, so she didn't have a ton of opportunities to actually do hands-on research, but she did serve as a peer mentor for introductory biology, and she was able to study abroad in Hawaii, where I think she was probably first introduced to coral reef ecosystems. She applied for an REU program through the Ocean Sciences program at CSUMB and was placed with Dr. Steve Palumbi at Stanford University for 2 summers, actually. The first summer was virtual and the second summer was in person. And she started work on corals at that time, exploring the resistance and recovery of Acropora hyacinthus corals in Palau. And I, at this time, was also recruiting Katrina to our lab to study very similar questions, but on corals in the Galápagos Islands. And Katrina joined the Moss Landing program and my lab in 2021. And the thesis research topic that you'll be hearing about in a few minutes is molecular mechanisms underlying heat and cold tolerance in Galápagos Pocillopora coral communities. Okay, so moving back to some of the fun stuff. So this is an AI-generated image that was inspired by Katrina. I put in some information into Gemini to produce this lovely image of Katrina moving to California from Long Island in combat boots. I also asked it to make her like walking quickly, but she kind of looks like she's just strolling down the road. So anyways, you're going to see some AI-produced images in Katrina's talk today as well. She made fast friends with the Moss Landing community, who are like-minded in the sense that you all love to dress up as well, so she fit right in. In fact, most of the pictures that I got from her peers were of costume parties. So here's some great images. She continues this tradition now that she's moved back to the East Coast with one of our former lab members up there, Ari, who is attending Katrina's recent birthday party and continuing to dress up, I think in this case, as Katrina's closet. And Katrina always brings with her a little bit of taste of her Italian heritage. She is a wonderful cook, and if you've had the chance to enjoy some of her cooking, you know how good it is, and she also just loves to do it. And she brings that joy with her wherever she goes. Okay, so on to some of her accolades. Katrina is an amazing teacher and mentor. Actually, two of her mentees are here today. Thank you, Kenzie and Matt, for joining us. You'll see in her talk today, too, she just has a real knack for explaining complicated concepts using analogies to broad audiences. So she was a TA in our introductory biology series in marine ecology, as well as sort of a de facto TA for my marine experimental physiology course for 2 years. And since she moved back to Long Island, she's also served as an instructor for the Cold Springs Harbor DNA Learning Center for middle school and high school students. She's a fantastic teacher, and she always meets students where they are. In terms of mentoring, she mentored 4 students in research and then another student through sort of a professional development program. So 4 of these students are shown here. We have Matt, Chris, Hannah, and Kenzie. Matt was actually a community college student who was doing the REU program at CSUMB and then transferred to CSUMB after that program and is now a master's student in our environmental science program. You can see that she brought these students all the way to presentations. So 2 of these pictures are from Mackenzie and Matt presenting their work. And she's just a really inspiring and caring mentor. And I'm so glad that you guys were able to make it here today. Katrina also traveled a lot more than many of my students. And although she never made it to the Galápagos, she did make it many other places around the world through her presentations and other activities. So these are all the many conferences that she attended, both regional, national, and international. I'll highlight 2 of them. One is the ASLO Aquatic Sciences meeting in Mallorca, Spain, where she was joined by Matt Bierman, her mentee, and they both presented posters at that meeting. And then also the European Coral Reef Symposium that was in Naples, Italy. So she made it back to her homeland as part of her science, too. She's also had many awards, which shows just what a wonderful grant writer she is. She was awarded the NSF IRES Track 2 Graduate Researcher Award, I guess. It's a program that brought her to Australia for 3 weeks, where she learned about coral reef ecology and dove on the Great Barrier Reef. She won 3 different Moss Landing scholarships, and the Myers Oceanographic and Marine Biology Trust, and then 2 pretty competitive CSU-wide research awards, the Sea Superb and the Coast Graduate Student Awards. Okay, so I'm going to leave you with this image here of Katrina, which just kind of encapsulates Katrina. She has such a zest for life. We really enjoyed having her as a lab member and loved her spirit in the lab. And even when she moved back to Long Island, she really was still a great lab mate and participant in the lab and helped out students in many ways from afar, so we really appreciate that. So good luck, Katrina. We're going to be sad to see you go, but we're also really excited to hear you talk about your research today. So welcome up. Thank you.
All right. I don't know how to get back to. Oh, that was really convenient. Just 1 second. Uh-oh. Okay. All right. Everyone can hear me? Amazing. So Hello, everyone. Thank you for coming to my thesis defense titled Molecular Mechanisms Underlying Heat and Cold Tolerance in Galápagos Pocillopora Communities. Coral reefs are often referred to as the rainforests of the sea. This is because, like rainforests, they are hotspots of biodiversity and provide many benefits to the organisms residing within. In corals, this can include nursery grounds, shelter and protection, hunting grounds, and food. They also provide many benefits to humans. They reduce wave energy by up to 97% in the areas where they reside, protecting coastlines from storm surges and erosion. It's estimated that through these services, they save the United States $1.8 billion a year in averted damages. They also support livelihoods through fishing and through ecotourism, and it's been estimated that through all of their ecosystem services, they can generate $3.4 billion a year in ecosystem value in the United States. Unfortunately, however, these benefits are threatened by the increasing prevalence of mass coral bleaching and mass coral mortality events. So before I get into what bleaching is, I want to talk about what a coral is. So this coral colony is made up of a lot of different coral polyps. So each of these is an individual coral animal in the phylum Cnidaria. So they're very closely related to jellyfish. And like all animals, corals need to eat. So they actually have tentacles that reach out into the water column and pull in food. But this is not their only source of energy, and it's not even their main source of energy. Corals have clear tissue, and inside are symbiotic algae. These algae provide coral with food from doing photosynthesis, and in return, the coral provides them with a home and some other amenities. But if the environment becomes too stressful, if it becomes too cold, too hot, too acidic, the algae stop doing photosynthesis as well, and ever the gracious landlord, the coral host says, hey man, you're not paying your rent, and starts kicking the algae out of its tissue, which leads to something like this. So that's that clear tissue with no algae inside and the white skeleton underneath. So this is a bleached coral, and although this coral is not dead, these algae can supply up to 95% of coral's energy requirements, so this coral is very likely to die due to starvation or disease. Coral bleaching is happening worldwide, as can be seen in this figure by Sully et al. So many reefs around the world have actually experienced 100% bleaching at some point in time, as shown by those yellow dots. And this bleaching can be caused by a variety of factors, but the number one most prominent cause of mass coral bleaching and mass coral mortality has been abnormal hot waters. So in this figure, we can see that after the 1970s, they started monitoring coral reefs after they started seeing bleaching. And we can see that coral reef cover, or hard coral cover throughout the world was pretty stable for a while, until a heating event in 1997 to 1998 led to the loss of 8% of all the world's corals. This is equivalent to all of the corals in the Caribbean Sea today. We then saw another heating event in 2010 and another in 2014 to 2017, and although I don't have mortality data for them specifically, we know that from 2019 to 18, we lost 14% of corals around the world, which is equivalent to all the corals in Australia. If I extended this timeline to today, we would see that we are currently in another heating event. And again, although I don't have mortality data for this, I know that in our past biggest event, 68% of reef area around the world experienced bleaching level heat stress, where in this event, 84% of reef area around the world has experienced bleaching level heat stress, with over 83 countries reporting signs of mass coral bleaching. So what can be done to save our reefs? Coral scientists have tried to maintain ecosystem function and preserve genetic diversity by initiating reef restoration projects. So in these projects, they grow corals in sites where reefs have been degraded, and then they transplant healthy individuals onto degraded sites. But these projects, in terms of time, money, and energy, are very expensive. So we want to make sure that a stress event is not just going to kill off these corals once another one inevitably comes through. It's been shown that we can increase the success of these projects by identifying stress-tolerant individuals for transplantation. But where do we find stress-tolerant individuals? Marginal populations, which are populations of species located at the edge of their environmental tolerances or limits, are generally adapted to suboptimal conditions and more frequent and intense stress compared to core populations. It's thus been suggested that they may host stress-resilient individuals. In looking for marginal coral populations, we turn our attention to the eastern tropical Pacific Ocean, specifically the Galapagos Islands. So Galapagos corals, or in general, a lot of corals prefer nutrient-poor, warm, and stable conditions. But Galapagos corals experience a cold water current coming up from the Antarctic and a deep sea current that brings in nutrient-rich cold water, making this southwestern region of the archipelago generally cold and very variable in terms of sea surface temperature throughout the year. A warm water current coming down from the north leads to the far northern region staying relatively stable but being warmer throughout the year. In addition to these variable sea surface temperatures, some other suboptimal conditions these corals are adapted to are relatively acidic waters, relatively low aragonite saturation, which can stunt coral growth, high bioerosion, and high nutrient levels, which can lead to overgrowth by external algae. These corals are also frequented with more intense and frequent stress events. In this figure adapted from Glynn et al. 2018, in the top left, we are looking at sea surface temperature, which you can see rises and falls every year. And on the right, we are looking at accumulated stress. So this is stress measured as the number of weeks corals experienced relatively high in the red box temperatures or relatively cold in those blue box temperatures. And in 1982 to 1983, there was an El Nino event that led to the loss of 97 to 99% of all the corals in the Galapagos. In 1997 to 98, there was another strong El Nino event that led to the loss of 26% of what was left. And in 2007, there was a strong La Nina event that led to cold water bleaching and mortality throughout the archipelago. So, I had said before that marginal populations generally hold stress-resilient individuals. Was this the case for the Galapagos? This figure is showing all the coral reef communities and all the structural reefs that existed prior to that first big El Nino event. Since that El Nino event, we have lost quite a few coral communities throughout the archipelago, and we've either seen the degradation or the loss of all of the structural reefs except for one. And if you turn your attention to that far northern site that includes Darwin and Wolf, you will see that far northern corals have exhibited the highest coral community resistance and recovery to stress, and Darwin holds the only remaining structural reef in the region. And in 2019, experimental bleaching assays found that cauliflower or Pocillopora corals had the highest heat and cold tolerances in those far northern sites when compared to other sites. And I actually want to focus in on this last bullet because these experiments are where I got my samples from. So sampling in 2019 was done by a team including my primary advisor, Dr. Cheryl Logan from CSU Monterey Bay, Dr. Margarita Brandt from Universidad San Francisco de Quito, and then other collaborators, Caroline Rodriguez, Steve Ryan, and Daniel Barshiz. And what they did was they assessed the bleaching tolerance of corals from 7 sites representing the thermal range experienced by corals in the region. And although they didn't only study Pocillopora or only assess Pocillopora corals, I chose them as my study species as they have a lot of those characteristics that are very relevant with marginal populations. They are well adapted to disturbed and productive environments, and they are key framework builders in areas where other corals struggle to establish. They're also incredibly important to the Eastern Tropical Pacific. They're the most prominent source of calcium carbonate in the ETP, and they're one of the only genuses found throughout the entire Galapagos archipelago. So what they did was they sampled 8 parent colonies from each site. And later on, when I say genotypes, I'm referring back to these original parent colonies. They fragmented these colonies, and they took small fragments and assessed those fragments' response to bleaching tolerance using the Coral Bleaching Autonomous Stress System, or CBAS. And what CBAS is, it has tanks that you put water from the local ecosystem and coral fragments in, and then you either rapidly increase or rapidly decrease the temperature in the tank, and then slowly let it come back to normal ambient temperature, mimicking a heat or cold stress event. On the day of collection, they did experiments for heat stress. So they had one tank that had normal Galapagos seawater temperature, 26 degrees Celsius, which represented our control. And they had another tank that went all the way up to 34 degrees Celsius, representing heat stress. And they assessed tolerance of one genotype, one fragment per genotype. They also, on the second day, assessed cold tolerance, again using the same control temperature, but ramping all the way down to 12 degrees Celsius in this experiment. And they measured tolerance as change in FV over FM, where FV over FM is a measure of how efficiently photosystem 2 converts light energy into chemical energy. It's therefore just basically a fancy way of saying it's a measure of photosynthetic health. And they measured changes in photosynthetic health between control-treated colonies and stress-treated colonies. And here are their results. So if we look in this top left of this figure, this is change in FV over FM. This is where the change is 0, where there was no change in photosynthetic health. And you can see that our control samples averaged around 0, which is what we would expect. But our heat and cold treated samples, if you see larger changes in photosynthetic health, that means that colonies from those sites had lower stress tolerance to that stressor, where smaller changes means that those colonies had greater tolerance to those stressors. And in reference to all the sampling sites, they found that treatment and site drove thermal tolerances, with Pocillopora colonies from Darwin and Wolf having the relatively greatest heat and cold tolerances and significantly greater heat and cold tolerances when compared to colonies from Isabella. So my thesis is looking at what is driving these regional differences in tolerance. And it's very interesting that Darwin and Wolf are both heat and cold tolerant, considering, as you can see, they never really experience cold water. So I hypothesized that 3 different things are driving these differences: MTORF haplotype, symbiont community composition, and gene expression patterns. I'm going to go through what each of these are. So traditionally, we identify Pocillopora species using the way that they look, or morphology. And if I was looking at these 2 pictures, I would think they were the same species, and I would be wrong. So this is called a cryptic species, or different species that look the same, and they are very prevalent throughout the Pocillopora genus. If I looked at these 2 corals, I might think that they're different, and I'd be right, but I would not guess that they are the same as the coral above them. This is called a polymorphic species, or a species with multiple morphs or forms, and they are also very prevalent throughout the Pocillopora genus. So instead of identifying Pocillopora corals using morphology, we instead identify them using their mitochondrial DNA, specifically the mitochondrial open reading frame sequence, or the MTORF sequence. And instead of saying different Pocillopora species, we say different Pocillopora haplotypes. 2 haplotypes are known to exist throughout the Galapagos: type 1 Pocillopora and type 3. And I'm going to come back to them in just a second. I also hypothesized that symbiont community composition would drive these regional differences, because if you have more tolerant symbiotic algae in your tissues and a stress event comes through, they're not going to stop doing photosynthesis, and the coral host is not going to kick them out. There are 2 symbiont species known to exist in Pocillopora throughout the Galapagos, and that is Durastinium species and Cladocopium symbiont species. And Durastinium species are relatively more heat and cold tolerant compared to Cladocopium. And it's been suggested that type 1 Pocillopora is more heat and cold tolerant than type 3 because it has a greater affinity for Durastinium symbionts than type 3 does. In fact, in the eastern tropical and continental Pacific, most often type 1 Pocillopora, but always with Durastinium symbionts, tend to have the highest stress tolerances after big stress events. My third hypothesis was that gene expression patterns might be driving these differences. So now we're talking about our nuclear DNA, and gene expression can be kind of a confusing thing to understand, so I'm going to try to break it down in a more simple way. So all of our DNA in our cell or our genome is kind of like a cookbook. And you don't ever try to make every recipe in a cookbook all at the same time. You choose one recipe at a time. So I want you to imagine that our cell is a commercial kitchen. And every time someone orders a meal, the chef has to go into the office, which is our nucleus. It has to open up the cookbook to just the recipe it's interested in. And he doesn't want to take the recipe book out into the kitchen. It'll get dirty. So he copies down just the recipe he's interested in onto a separate piece of paper. This is the process of transcription, where a gene that codes for a certain protein is transcribed into mRNA. This mRNA transcript will then go out into the cell, where it will get cooked up or translated into our final product, a protein. So when we say that we're measuring gene expression, what we are measuring is the expression of proteins by measuring how many transcripts of genes were present in those samples. So if I had 5 transcripts, is that a lot or a little? Who's to say? The thing with gene expression studies is they have to be comparative. So if I had one coral that I found that they had 5 transcripts of a gene, and another expressing over 40 transcripts of a gene, I would know that the top was relatively low expression and the bottom was relatively high. So in terms of my samples, whatever they were expressing at that control temperature is our baseline for gene expression. And if, let's say, genotype 1 went from expressing 6 transcripts to 10, this is called upregulation, or an increase in gene expression. Let's say genotype 3 went from expressing 3 transcripts to 2. This is called downregulation, or a decrease in gene expression. Acute heat and cold stress have induced differential expression, or significant up or significant downregulation in many genes that have now been linked to stress. But before I get into that, I kind of want to talk about what physiological changes are associated with these stressors. So when corals are exposed to heat stress, things start to really ramp up in their systems and proteins start to unfold. There's a lot of oxidative stress, which makes byproducts that are very harmful to the coral, and so on. Cold stress actually slows things down. It makes energetically expensive processes like growth and metabolism slow down. And although we have a lot of studies that have looked at these physiological differences between heat and cold responses, we do not really have many studies on heat versus cold responses of gene expression in coral. And gene expression is really interesting because we can see these processes change before they actually physically change by looking at their expression. So overall, we've seen an activation or an upregulation after stress of genes relating to chaperones. So chaperones are proteins that refold proteins that have been unfolded after stress, especially heat shock proteins. Antioxidants, which remove harmful byproducts of oxidative stress. Cell death and immune signaling genes, and cell structural regulators. We've also seen a downregulation of genes related to growth, development, and metabolism. But this data is really mostly from heat stress studies. There have not been many that look at cold stress and not many that compare heat and cold stress. So comparing these corals' responses to heat and cold stress was really important to me because, as I said before, corals in the Galápagos are experiencing bouts of stressful cold temperatures. So in order to test my hypotheses, I analyzed the haplotype and symbiont composition of 5 samples from all 7 sites with an extra 5 samples from Darwin. And while my intention was to analyze gene expression in 5 heat and 5 cold-treated samples plus their respective controls from all 7 sites, unfortunately, due to low RNA quality and financial constraints, I could not do that. But instead, I looked at gene expression in 5 heat and 3 cold-treated samples plus their respective controls from only Isabella. So Isabella was that site with really low cold tolerance and heat tolerance. And the most interesting thing about Isabella is our colonies were sampled from a population previously determined to be comprised of completely genetic clones. So what this did is it increased my sample size and replication within the site for comparing those heat versus cold stress responses. So before I move on, I just want to clarify that instead of the traditional methods results discussion of a defense, I will be following that flow, but within haplotype and symbiont community analysis, and then within gene expression pattern analysis, just for clarity. So in order to analyze haplotypes, I extracted DNA from 5 control-treated samples per site from the day of collection. I chose control-treated samples and the day of collection to try to most closely represent the symbionts that were present in those samples prior to collection. I then amplified the Pocillopora MTORF sequence using polymerase chain reaction from these samples and sequenced using Sanger sequencing. I aligned these sequences to published MTORF types and assigned haplotype based on which one was the best match. And onto my symbiont community methods, I used the same DNA that I extracted from my MTORF haplotype analysis, except instead of amplifying the MTORF sequence, I used quantitative PCR to quantify the relative amount of durstinium, those stress-tolerant symbionts, and stress-sensitive cladocopium symbionts in those colonies. So what I found was that all 5 samples from Isabella contained only stress-sensitive cladocopium symbionts. And this was not only the case in Isabella, but in Santa Cruz, Floriana, and Espanola, and for 3 colonies from San Cristobal. The other 2 colonies from San Cristobal contained increasing amounts of those stress-tolerant durstinium symbionts. And then when I looked at those very resilient far northern colonies, what I found was they contained the greatest amounts of durstinium symbionts compared to my other sites. And a preliminary ANOVA test did not find that there was a significant relationship between symbiont composition and tolerance, but I will be repeating that with my full data set soon. When looking at MTORF haplotypes, I found that all of my samples from Isabella and Santa Cruz were MTORF type 3. And genomic testing by a collaborator found that all 5 samples within sites were genetic clones of one another. All the rest of my samples from the other sites were MTORF type 1. And I'd like to just highlight what's really interesting about these results. So we found that my heat and cold tolerant colonies from the far north were MTORF type 1 and hosted the greatest amounts of durstinium symbionts, while my heat and cold sensitive colonies from Isabella were MTORF type 3 and hosted only stress-sensitive cladocopium symbionts. And I'm going to come back to this data in a minute, but first I'd like, oh, no I'm not. I'm going to talk about it right now. So this data is actually a great stepping stone in data that's been tracking Pocillopora communities in the Galapagos through all these stress events. So we have data from 1998, 2012, and now 2019. So that study in 2012 found type 1 Pocillopora in Darwin and Wolf, and type 3 genetic clones in Isabella with only C symbionts, those stress-sensitive symbionts. And both of these matched our results. However, ours is the first study to quantify symbiont community in those resilient far northern sites. It's also the first to directly relate these characteristics to experimentally derived heat and cold tolerances rather than just extrapolating heat and cold tolerance based on post-bleaching survival in the field. This other study found type 1 Pocillopora in Santa Cruz and Floriana with mixed symbiont communities, which did not at all follow our results. We found clonal type 3 colonies in Santa Cruz and only type C symbionts in Santa Cruz and Floriana. This may reflect a shift in community structure in Pocillopora communities in the Galapagos, and because these populations are marginal and are experiencing conditions expected to be experienced by reefs worldwide in the face of climate change, this shift can offer insight into how Pocillopora communities could shift across their range. Now I will be coming back to this data in a bit. So I'd like to move on to my gene expression analysis. So I extracted RNA from 5 heat-treated and 3 cold-treated colonies, plus their respective controls from just Isabella. So again, that was the very stress-sensitive site. I converted RNA, which, like a piece of paper, is very unstable, into stable cDNA and sequenced using Illumina HiSeq. What I was left with were millions and millions of copies of transcripts that look something like this. So the first thing I did was performed quality control using FastQC and Trim-O-Matic, trimming out leading and lagging unassigned bases, and also trimming out any unassigned bases within the transcript. And then if the transcript was too short to be read, the program dismissed it. I then annotated these transcripts using searches against a database, essentially looking up the content of the transcript and annotating what meal it was a recipe for, or what protein it was a recipe for. I aligned these transcripts to the published Pocillopora dammicornis genome published by Cunning et al. 2018, essentially figuring out where all of these transcripts align to in the genome, and quantified and consolidated transcript abundance using Salmon and TX import, figuring out how many orders there were for each protein at the time the samples were taken. And what I found, and I should clarify that these are not all of my gene expression results, but the most interesting ones. I first did principal component analysis of all the gene expression data before I did any kind of differential expression analysis. What this PCA plot is going to show is how similar different colonies' expression patterns were. So if it looked something like this, which it did not, that would mean, I'm going up here, that would mean that samples' genotypes had more similar expression patterns than within treatment. But we did not find that. We found that samples very clearly separated by treatment, with control on the left and heat-treated samples on the right. And PC1, which separated these samples by treatment, explained a relatively higher amount of the variance observed in this plot. And for reference, this is a pretty high correlation value, or PCA value, for data prior to differential gene expression analysis. We saw the same thing happen with my cold data set, where samples, again, treatment drove gene expression patterns more strongly than genotype. I then conducted differential gene expression analysis with EDGE-R, which allowed me to see which genes were being highly upregulated or significantly downregulated. And before I get into the resulting heat map, I want to go over what we're looking at. So each column represents one sample. In this case, genotype 1, that was heat stressed. And each row, each very, very skinny row, represents one gene. The color of that row represents its expression, with red being relatively high expression and blue being relatively low. And when I say relatively, it's because I'm comparing it to its control. So all of these genes had relatively low expression in the control sample and relatively high expression in the heat stressed, meaning they were upregulated. And we found that there were 348 genes that were differentially expressed after heat stress. And among them, they were generally balanced in terms of upregulated and downregulated genes. In both my heat and my cold stressed experiments, we saw that, again, colonies grouped by treatment rather than genotype. And in my cold stress experiment, I found 202 genes differentially expressed after cold stress. And the broad majority of them were downregulated, which again was expected because cold stress generally leads to a slowdown of a lot of main processes. When I looked into these lists of differentially expressed genes, I found that 82 of them were in common between these two stressors, which was very interesting to me because corals have been going through heat stress events for kind of a while now, but a lot of scientists have been concerned that increased heat resilience will come at the cost of decreased cold, and I'm kind of interested to see why these corals, or what genes, are being differentially expressed in response to both treatments. A PCA plot of this showed something very interesting, where again, these samples separated by treatment rather than genotype. And what that means, if I can do this, what that means is this heat-treated sample over here had more similar gene expression patterns to these three cold-treated samples than to these three heat-treated samples. And that was supported by a heat map, again, which grouped samples together by treatment. When I looked into what these genes were actually doing, I found that The upregulated genes were generally related to cell structure and cell signaling, which is missing a few categories that I thought would be upregulated, which I'll get into in a second. Whereas the broad majority of them were downregulated, and they were related to protein quality control and turnover, metabolic enzymes, growth and development, redox and metabolic stress response, and cell signaling. So I expected protein quality control and redox and metabolic stress enzymes to be upregulated. So I was very confused as to why they were in the downregulated group. So what I did next was I looked at the differentially expressed genes that were specific to just heat or just cold responses. And what I actually found was many of them fell into the same gene family or the same functional group. So they really actually weren't separate responses. And they fell into the same functional groups as many of those shared genes. And I know that this is a lot to look at, so I'm going to kind of condense it. So what I found was there was a very apparent lack of upregulated cold response genes in these functional groups. But those genes that I expected to be upregulated in response to stress were, but they were only upregulated in response to heat stress. I also found that one functional group that has been widely studied was not represented in both heat and cold responses, and that were chaperones. They were only represented in heat responses, and all of the chaperone proteins I found were upregulated, including 2 heat shock proteins. Again, this makes sense, as heat generally induces that unfolding that chaperones would fix. So to summarize, I found that treatment drove gene expression patterns more strongly than genotype or experiment. I found a heat-specific activation of chaperones and heat shock proteins. And I found that colonies exhibited a shared environmental stress response to both heat and cold stress, with about a quarter of heat DEGs being shared with cold response and almost half of cold DEGs being shared with heat response. And many of those unique genes, again, fell into the same functional categories. And this is really interesting because this may explain why far northern corals are more tolerant to both heat and cold stress, even though they're not experiencing cold stress. It might just be because they have the same response to both stressors. So when I looked at other papers, I found that broadly my results followed what other papers found. But I found something very, very interesting when it came to that shared environmental stress response. So this ESR was first described in yeast and later in many organisms, including 2 corals. And what they found were that the same genes shift in expression in response to both heat and cold stress, but how they shift depends on the severity of the stress. High-intensity stress tends to activate protective and stress response proteins, while it tends to shut down those energetically expensive ones. Whereas mild or chronic stress tends to suppress both groups. When it came to my data, I found that my heat response genes generally followed this high-intensity stress response, with upregulated genes that were related to heat response and protection, and downregulated genes related to growth, development, and metabolism. My shared and cold response genes generally followed this mild or chronic stress response, with both categories in the downregulated genes, represented in the downregulated genes. This is and why this is so interesting to me is although my sample size was too small to do any kind of reliable statistical analysis to compare gene expression patterns with tolerance, my patterns indicate that my colonies would have a higher cold tolerance and a lower heat tolerance, as cold induced a mild stress response and heat induced a strong stress response. And that actually matched our data. We found that colonies from Isabella had greater changes in photosynthetic health in response to heat rather than cold stress. And this kind of makes sense because samples from Isabella, or colonies in Isabella, are generally exposed to more cold temperatures than they are to heat. This represents the first gene expression study ever done in Galapagos corals. It is also one of the only studies that have compared both heat and cold stress responses in corals, and 2 of these other studies have also found that corals may rely on a single conserved stress response system rather than separate pathways for heat and cold stress responses. But mine is the first to directly compare thermal tolerance measurements with these expression patterns. And what my data shows is that tolerance might be reflected by how colonies deploy the stress response system rather than whether the stress is heat or cold. So again, this might explain why samples from Darwin and Wolf are more heat and cold tolerant, even though they're never exposed really to cold waters. So to bring it back to my hypotheses, I expected that MTORF haplotype, symbiont community composition, and gene expression patterns would explain these regional differences in stress tolerance. And my gene expression patterns generally followed this because although Isabella colonies were generally sensitive to both stressors, they were more tolerant with insight to cold than heat, which was consistent with their gene expression patterns. However, I would like to redo this analysis with full statistical testing from tag-seq RNA data from all sites to see if maybe Darwin and Wolf will support this finding. In terms of MTORF haplotype and symbiont community composition, again, I found that my hypotheses were supported mostly, with heat and cold tolerant colonies from the far north being MTORF type 1 and hosting the greatest amounts of D symbionts, and heat and cold sensitive colonies from Isabella being MTORF type 3 and hosting only stress sensitive cladocopium symbionts. The implications for this study, the Galapagos have conditions that, again, are expected to be experienced by reefs worldwide in the face of climate change and recurrent heat and cold stress events. So understanding mechanisms of resilience here may help us to understand mechanisms of resilience elsewhere in response to both heat and cold. Understanding differences in heat and cold stress response is especially important in the eastern tropical Pacific and the Florida Reef Tract, where corals are generally, even though waters are warming, these corals do experience bouts of cold stress, generally due to La Nina events. And finally, my findings can guide restoration projects by elucidating characteristics of stress tolerance to increase the success of restoration projects. And in fact, they already have guided some restoration decisions for a restoration project that is being done in Isabella. And with that, I would like to thank my primary advisor, Dr. Cheryl Logan, for her guidance. She has been absolutely imperative to my whole thesis, not just because she's my advisor, but because she has been so comforting and kind to me throughout the whole process. I'd like to also thank my co-advisor, Dr. Amanda Kahn, who again was very emotionally supportive of me, especially when I was first learning how to do molecular techniques. And her molecular techniques class is actually a lot of what set me up and my thesis for not just my thesis, but for my job at the DNA Learning Center. I'd also like to thank my committee members, Dr. Scott Hamilton and Dr. Nathaniel Jew, for their help and their classes, which taught me a lot about statistical analysis in terms of ecological studies and bioinformatic analysis that was imperative to my thesis. I'd also like to thank the Logan Lab, which is not fully represented by this picture, but thank you guys so much for always being so supportive. And I'd like to highlight a thank you to Ari Dash. Ari Dash sat with me on Zoom and in person for countless hours, week after week, helping me create my bioinformatics pipeline code line by line. And he didn't have to do that, and I really, really appreciate that. And because I didn't get the chance to send in embarrassing photos of him last year, or when he did his defense, I would like to highlight that last year when Ari came to my birthday party, I told him that the theme was either going to be by the sea or wear all black. And Ari said, "Don't worry, you don't have to tell me. I have something for both." And he showed up in the fall in New York in a wetsuit to my birthday party. The following year, when the theme was selfishly to dress as myself, or to dress like myself, he came as my closet. So thank you, Ari, for your laughs and your help. I'd also like to thank my mentees, Hannah Kim, Matthew Bierman, Christopher Shinoy, and Mackenzie Caprino. I literally could not have done this thesis without you guys. You were in the lab with me for countless hours, extracting and re-extracting, and then re-extracting again DNA and RNA from these samples. And I cannot wait to see what you bring to this world. You guys are going to be so successful, and I'm so proud of you. I'd also, of course, like to thank the sampling team, and I'd like to highlight a thanks to Dr. Margarita Brandt and the Galapagos Science Center for doing all the restoration and caring so deeply about the reefs in the Galapagos. And I'd like to acknowledge our collaborator, Dr. Michael Connolly, who is the one who found that there was those, through genetic testing, those clonal genotypes in Isabella and Santa Cruz. And of course, I would not have been able to do any of this work without very generous funding from these sources. And finally, I'd like to thank my friends. I could not fit you all on one page, and for that, I am actually truly blessed. If you're watching at home and you're not up here, I'm sorry. But you guys have been an incredible support system throughout these past few years. And I'd like to really highlight at the end my boyfriend, Joseph, who drove all the way out from New York with me for me to defend my thesis. It has been imperative to my mental health these past few months. And finally, I'd like to thank my family, who again is not all pictured here because that would actually bring us all the way back to Italy. I'd like to thank you guys. You have been so supportive of me through this whole process, and you've also really encouraged me to, as Cheryl said, keep getting up, even though things tend to try to tear me down, but they can't because I have such a strong support system here. And with that, I thank you guys for listening, and I will take any questions.
Wonderful job, Katrina.
Thank you.
Does anybody have any questions?
Okay. Yeah, I guess just in general, with the coral colonies that you looked at from Wolf and Isabella, you said most of them were wiped out and these are the newer colonies, or was there like a specific age of the colony, like between the two that you saw more cladocopium or Dirtostinium in?
So sampling was done randomly, but in Wolf, generally a lot of those were probably older colonies because they have had the highest resistant to stress events, whereas all of the colonies sampled from Isabella were taken from a colony that Baums et al. 2012 found that all of the colonies in this population originated from one colony breaking off and regrowing this population. So they were all generally new, relatively new, 2012, and relatively new, and they were all genetic clones, whereas in Wolf and Darwin, we saw a lot more genetic diversity, and probably those colonies were a bit older.
Thank you.
Of course, thank you.
Great job, Katrina. Do you think that it would be possible, knowing the currents of the Galapagos, that there's any sort of dispersion of larvae between the islands, or do you think all of these genetic differences are driven by more evolution?
So modeling has actually shown that within those far northern sites of Darwin and Wolf, there is a lot of migration going on in terms of genetics. There's a lot of flow between the two sites, but they do not flow down to those central southern sites. And those central southern sites can flow up, but again, not down. So that's why there's a lot more diversity in those northern sites, which may, as we know, diversity tends to lead to greater amounts of tolerance to variable stress. And that might also be a factor of truth.
Fantastic job, Katrina. I love that presentation.
Thank you.
Thinking about the Darwin, you weren't able to do the gene expression analysis of any of the Darwin and Wolf corals, but can you speculate what you think the patterns would look like, knowing that they are more tolerant, right, to heat and cold stress. Do you think there's be less differential expression overall, or there maybe more expression of protective genes?
Yeah, so I think that if I compare them with insight, I might see less, in general, less differentially expressed genes overall, just because they're not responding as strongly to these stressors as seen by their greater heat and cold tolerances. Or I might see that mild or chronic stress response where there was a broad downregulation of a lot of genes rather than an upregulation of stress-specific responses. However, other studies have shown that heat shock proteins and other stress response genes have been upregulated in more tolerant colonies. But again, we generally saw that within Isabella, more tolerant colonies had that broader suppression. So I'm not positive, but I would speculate that they might follow the cold response from Isabella more closely. But if I compared Isabella to the far north, I would expect that the samples from Isabella will now show something more like that high intensity response, whereas Darwin and Wolf might show that more low intensity response.
Amanda. And also, if there's anyone online on Zoom, I don't know how we gain. No, there's not right now. Okay.
Thank you. Scott took my question, which is great. I think it's a really cool question. So I want to follow on that with
Yeah.
the thought experiment thing.
Yeah.
So this was based on the experimental design, the immediate response of the corals
Yes.
to this stress. So I guess in a two-part way, which of the gene expression responses do you think might have been related to the act of bleaching? Like, I don't know what a coral needs to do if like immune stuff is upregulated or how they decide it's time to boot out the symbionts. And then, I mean, from other studies or just from what you know and what you've seen, what do you think would happen if left in those treatments for longer?
So chronic stress, well, actually I can speak on the fact that 34 was actually not the highest temperature we subjected these samples to. 35.5 degrees was, but because it caused such great levels of bleaching, it was really hard to get good quality RNA from them. So when left to much more chronic responses, I'm not sure how well we could extract RNA for that type of analysis. But generally, when samples are left with chronic responses, we see an opposite of the gene expression patterns seen in acute stress response, which can make it very confusing when you're looking at gene expression data in other studies, because you'll be like, oh my goodness, this is the exact opposite of what I found. And then you'll look farther and find that they stressed their samples for much longer than you did, or they took their samples from in situ stressed after a bleaching event rather than experimentally stressing them.
Very interesting.
Thank you.
Thanks.
Okay, any other questions? The whole committee.
Whole committee. Sneak preview into the next half hour.
That was a great talk. Really nice job, Katrina. This is not a difficult question. I was just kind of curious. You mentioned at the end that they'd been using some of your data for restoration efforts. I wonder if you could talk a little bit more about that. I was curious to know how they were using it.
Yes, so from my understanding of it, it's not necessarily what I have found that they've been using, but they used the data from those CBAS assays and the tolerance of which genotypes were more tolerant than others to inform decisions. So now we have another layer of data that we can add on to that. I mean, we showed that type 1 Pocillopora are the ones that tended to have those stress-tolerant Duracinium symbionts, and samples with Duracinium symbionts tended to have greater stress tolerance. So maybe it should be those characteristics that we are focusing on in the future for reef restoration. So in general, they chose the ones that had the greatest heat and cold tolerances for growing for restoration is what I would imagine. But now we have an extra layer of data to really pinpoint that. Thank you.
That's a nice sneak preview of Ava's thesis that she'll be defending later this year, or next year in May, hopefully. So I think, unless there's no more questions, I just also wanted to give a shout out to Katrina's parents who are joining online because they really wanted to be here, but because of the shutdown and flight cancellations, et cetera, they couldn't make it. So thank you To Susan and Andrew, also for sending the great photos that you saw earlier today. And with that, let's give Katrina one last round of applause.
Thank you. Okay, bye. Is Yuri still back there? Good job.